All right, well, welcome everybody. Welcome back to Flyway Nights. We've been on our hiatus here, but now it's fall again, and we are getting set. So I'm going to bring up our little screen here. There we are. So welcome to Flyway Nights, our 6 months out of the year. We do presentations on local topics having to do with the Central Valley and often with the Yola Bypass Wildlife Area. Tonight, we're very excited. We have Jackelyn, who is a PhD candidate here at UC Davis, and she's going to be sharing with us some of the issues that, of course, we're facing coming from our tires as we're driving around, and how that is affecting the fish in our delta. Next month, well, we're still working on it. We got some queries out, not real sure exactly what we're going to be doing, but we're hopeful. So keep your ears open, watch our website. If you get our email newsletter, you'll also get info about that. But I thought I'd also share with you what we have coming after December. So in January, we're going to be hearing about the burrowing owls. February, we're going to hear about river otters. And then in March, wildlife spectacles. So it's going to be a little bit of a variety of things having to do with the wildlife area. And then in April, we will hear from Garrett with his updates of the Yola Bypass Wildlife Area. So I am going to take my screen back down and pass over to Jackie so that she can share with you. And as in the past, if you have questions, we do ask that you go ahead and toss them into chat. And then we can read those out after Jackie has done her presentation. All right. So take it away, Jackie.
Let me just get this fully situated. All right. Does that look good on your end?
Looks great. Thanks.
Okay, perfect. So yeah, hi everybody. Yes, I'm Jackie and I'm a PhD student at UC Davis. I'm in the agricultural and environmental chemistry grad group. And today I will be talking about a project that I've been working on for about the past 2 years, related to a contaminant in tire rubber and how it's affecting our fish. But first, I thought I'd spend a little time introducing myself. I've been at UC Davis for quite a while. I did my bachelor's here as well and graduated in 2015. My bachelor's was in environmental toxicology, and so I was really focusing on aquatic toxicology, so how pollution is affecting aquatic ecosystems and all the different organisms that you might find in different ecosystems. And after that, I spent some time doing some consulting for an aquatic toxicology firm, actually based out of Fairfield. So we did work a lot in the Sacramento-San Joaquin Delta still. And in 2018, I came back to UC Davis as a staff researcher where I joined a lab where we were focusing on microplastic pollution. And so it's a really fun time because it was still a really hot new research topic. And when I started my PhD a couple of years later, I knew that it was going to have to be related to microplastics in some way. And so I spent my first like quarter-ish of school of my PhD really looking through the literature, trying to find a topic that I thought was so interesting that it could carry me through 5 years of study. And I was very lucky because in December of 2020, my favorite scientific paper ever was published. And this is the title. It's called "A Ubiquitous Tire Rubber Derived Chemical Induces Acute Mortality in Coho Salmon." And so the connection here is that rubber, as we drive down the road, it sheds little particles, and those are types of microplastics. And so I finally found my holy grail PhD research topic. And I really liked this paper because it was kind of, it kind of concluded a multi-decade, like almost murder mystery that we've been seeing. So I'm going to transport us out of the Sacramento-San Joaquin Delta area, and we're going to plop ourselves into Seattle, and especially the streams kind of near Seattle that are still considered urban streams where coho salmon swim upstream to their spawning grounds. And so for the past 3 decades, people up in Seattle have been seeing these weird mortality events in coho salmon where they're swimming upstream, and before they are able to spawn, they had these big mortality events, and the fish that were symptomatic looked like the one in this picture here. So what they were seeing is fish that are unable to kind of maintain their equilibrium. They're swimming all wonky. They're surface swimming and swimming in spirals. And then they were also kind of gasping more, and their gills were ventilating more frequently than we would normally expect. And so it took a lot of researchers many years to narrow down exactly what was happening. They basically figured out, well, it seems to be associated with water that's running off of roads during rain events. So they looked at all of the different kind of components of what could be adding pollution to roadways. So they looked at tires and brake pad wear, which can give off heavy metals. They looked at oil that can drip out of cars. And they figured out that it seems to be related to tire leachate. And so over time, they kind of started splitting tire leachates into different chemical fractions and testing those and seeing which one is still toxic and which one is not toxic. And they did that over and over and over again until they were able to narrow it down to this one chemical that has been causing all of this grief for the past 3 decades. And that chemical is called 6-PPD quinone. And 6-PPD quinone is not actually an ingredient in tires, but it's the transformation product of an ingredient in tires. So the ingredient is called 6-PPD. It's the chemical that's on the left side of this chemical equation here. And the giant scary word below it is actually the full name of 6-PPD, and I still don't know how to pronounce it, so I'm not going to try. But 6-PPD is a really important ingredient in tires because it protects the rubber from reacting with ozone. So we have ozone in the air kind of all the time, especially in the summer. And that ozone, it can attack rubber and cause cracking and crazing. It can cause really big safety issues because if you have cracks in your tire, it can cause a blowout while it's in use. And 6-PPD also really increases the longevity of our tires. So with 6-PPD, our tires last about 60,000 miles on average, but without 6-PPD, they only last about 5,000 miles. And so it's really helpful for making sure that we don't have to just burn through tires like crazy as well. And so because 6-PPD is so important to the safety and longevity of our tires, it's present in basically every commercial and passenger vehicle on the planet, in the tires on the planet. And it's funny because this reaction that the 6-PPD is designed to undergo, it's supposed to react with this ozone before the ozone reacts with the rubber. That reaction with the ozone forms this transformation product that is unfortunately very toxic. And so I just want to highlight, in fact, I can get a laser pointer out. Yeah. So the difference between 6-PPD and 6-PPD quinone is right here. So in 6-PPD quinone, we can see that it's got those 2 double bond oxygens, and that's the quinone structure. And quinones are pretty interesting because they are very reactive to chemicals and biomolecules. They can do a whole bunch of different things. And so because of that, we don't really know exactly what toxicity from a quinone is supposed to look like. There are some quinones that are naturally occurring in the environment that are not toxic at all. Some are actually good for us and are used in pharmaceuticals. And then, unfortunately, some like this one are toxic. But the main point here is just that just because it's a quinone, it doesn't really tell us a lot about what's actually happening in the fish to cause this toxicity. Okay, there we go. So if we look at this kind of chart on the left, we can see, you see a little road cartoon. So we've got cars rolling down the road, shedding tire wear particles. And then when it rains, those particles get washed into nearby urban streams. And so there's already some 6-PPD quinone in that roadway runoff before it hits the receiving water, or the stream where our fish are. And in those streams, it kind of gets diluted because there's so much water in the stream. And so in that original study that came out, they looked at roadway runoff concentrations, and they found somewhat high concentrations. They can be between 0.5 and 1.3 micrograms per liter. And then once it gets diluted into the stream, it reduces a bit, and the highest concentration they saw was about 0.23 micrograms per liter. However, the lethal concentration that people have measured for coho salmon is 0.095 micrograms per liter, which is extremely low, and much lower than what we're seeing in roadway runoff, and often lower than what we're seeing in receiving waters or the streams where our fish are. And so this measurement, we call it LC50. It stands for lethal concentration for 50% of the population. That is the concentration where we expect 50% of coho salmon to die and the other 50% to live. And it's a common kind of toxicity test that we use to be able to compare toxicities between chemicals and between species. And so also, luckily, that big study that came out happened to look at some of our urban streams in the San Francisco Bay Area. And so those detections are encircled in the red box. So if you look at the red dashed line, that is the LC50 or the lethal concentration for coho salmon. And if you look at the data for San Francisco, you see that the concentrations in those streams are right around that LC50. And sometimes there's even a few that are pretty significantly above that concentration. So we are expecting to see coho salmon mortality in our San Francisco streams based on the concentrations that we're finding. And so I again want to highlight that tire wear particles are a type of microplastic. And in the United States, we generate more tire wear particles per person on average because we live more dispersed, we drive longer distances, and we have bigger, heavier cars. And we really rely on trucking our goods around the country. And so we kind of have the problem a little bit worse than potentially some other countries. But in 2019, there was a big study based out of the San Francisco Bay where they were kind of dragging a net on the surface of the water and looking for microplastics in general. And what they found was that almost half of their recovered microplastics were black rubbery fragments that looked like tire wear particles. So it's estimated that, modeling studies suggest that tire wear particles are the biggest input of microplastics globally into the environment. And it's estimated that 36.2 million kilograms of tire wear particles are generated every year in the Bay Area. And if we assume that about 70% of that runs off when it rains, there's an equivalent to approximately 43,000 whole tires that are flowing into the San Francisco Bay every year in the form of those tire wear particles. And so with this slide, I really, what I want you to take away is that quantifying environmental tire wear particles is really, really challenging, and that has a lot to do with their size. So if you look at the photo on the right here, you see a tire wear particle that it's very zoomed in. This particle is about 15 micrometers in length, which is about the width of a pretty thin human hair. So very, very small. And this graph on the left came from data from a study where they were in the lab generating tire wear particles on a pavement simulator, and they were analyzing the size of the particles that were coming off. And so what we see is there's kind of two humps here. We've got one hump that's in about the 10 to 100 micron range, if you look at the x-axis, and then another one that's even smaller, kind of starting right around 10 and going lower to around 5 microns. And then there's this very sharp cutoff. And that is because we don't really have good methods in the lab for looking at particles that are smaller than 5 microns. It's really, really, really challenging. And so they just weren't able to do it. And so what we might expect is that if we had the full data set, we might expect that hump to kind of extend upwards and then also out to the left into those smaller particle size ranges into the nano size range. And this is problematic for us as researchers because traditional microplastic research is done using nets that have a mesh size of 333 microns. So that size range is reflected here in this kind of dark blue square. But luckily, that big microplastic study in the Bay, the San Francisco Bay, they were able to go a little bit lower, down to 125 microns, which is represented in this kind of lighter blue region. And if you look at where this orange size distribution of particles overlaps with what they were able to capture, you can see that it's a very, very small portion of the tire wear particles that are likely being generated and released into the environment. And so we think that because of these kind of discrepancies in our ability to measure and find tire wear particles and their tiny size, we think we're really underestimating the extent of contamination of tire wear particles in our aquatic ecosystems. So in 2019, 3 billion tires were produced globally, and in California, we had 51 million tires becoming waste every year. And so this is a chart that shows how those waste tires are managed. I got this from Cal Recycle. So this is the data from 2022. And we can see that about 50%, about half of the tires go directly to landfills. And then over here, you can see that some are retreaded or resold in the used tire market. And then the rest of these applications here are methods that we use for recycling tires. So, for example, sometimes we burn them to use them as fuel. But I want to highlight this kind of light purple pie slice here that says crumb and ground rubber, because that is one of the biggest uses of recycled tires, and it can be turned into a lot of different products. For example, we have started adding crumb rubber to asphalt to make it a little bit squishier. It's a little bit easier on our tires as we drive. And it's just kind of a good cheap filler. We also use the tiny crumbs to fill in artificial turf fields. So especially the sports fields that use those like kind of artificial green plastic blades, which by the way, we also find in aquatic ecosystems. But those blades are kind of infilled with all of this tire crumb so that it's nice and squishy. And then it's also used in children's playgrounds to make kind of that like soft rubber padding that the playgrounds are built on top of. And so the question here is really, are these recycled uses of our tires safe for us or for ecosystems? And so, for example, for asphalt, we don't really expect the tire particles to be mobilized because they're really stuck in that basically rock structure. And so we don't really expect that they're going to be released into the environment from the asphalt very easily. However, with those artificial turf fields, we play football on them and people run with cleats and disturb them and kick them up into the air. And then when it rains, a lot of those particles get washed into nearby streams. Those fields have to be replenished with tire crumb almost every year if it's like a pretty well-used field. And so all of that tire crumb is just being released into aquatic ecosystems, which is probably not great. And then again, for children's playgrounds, the squishy material that they play on is also kind of stuck, so we don't expect it to be released into the environment. However, it is concerning that kids are playing on these playgrounds when it's really hot and it's evaporating all those kind of tire chemicals that, including 6-PPD quinone, but also others as well. And so there's kind of a lot of questions around the safety of using tires for children's playgrounds. Okay, and so these images that I have here, I took from a review paper that was looking at all of the studies that had come out at the time, trying to detect 6-PPD quinone in different environmental matrices. So in this study, they found that people are detecting 6-PPD quinone across the United States, Canada, Europe, Asia, everywhere. And it's been found in fish that's sold at food markets. It's been found in human urine. It's been found in human spinal fluid. So it's really everywhere. But in these images, you can see all these little different types of like water samples. For example, we have urban runoff and stormwater here. And the number below, the percentage below, is indicating the percentage of positive detections in the samples in all of these studies that these authors were combing through. So we were finding 6-PPD quinone in 100% of urban runoff samples, 57% of stormwater samples. I won't go through all of them, but 6-PPD quinone is pretty water soluble. Well, I guess it's actually not that water soluble, but it's able to dissolve in water enough to surpass that LC50, that lethal concentration, because that lethal concentration is so low. So yeah, so we do find it in water, but we also are finding it in air and dust. It's not a volatile chemical, so we don't expect it to evaporate on hot days. So any 6-PPD quinone that we're finding in the air, we expect it to be sorbed or just stuck to the outside of a particle. So those particles could be just general atmospheric particles, dust, indoor dust, brake dust. So we do find it in air, and we are also finding it in soils and sediments. And there haven't been a lot of studies looking at it in soils and sediments, which I find a little bit concerning because it's a pretty hydrophobic chemical, meaning it doesn't really like the water, which is why it doesn't dissolve in it. And so instead, it's going to prefer to stick to soil and stick to sediments, and also to stick to biological tissues. So like it'll stick to our fish that are swimming through. And actually, the level of hydrophobicity that 6-PPD quinone has is kind of perfect for optimal uptake of the chemical across gill tissue. So it's actually makes it very bioavailable to our fish, and we do expect that the main route of exposure in fish is across the gills. And so since that big landmark study came out in 2020, toxicologists across the globe have been testing this chemical in a variety of different aquatic species. And the results are very odd, I find. There is basically no toxicity in any animal that's not a fish. So no invertebrates that have been tested seem very susceptible. Maybe some very mild things, but they're definitely not experiencing mortality. And even fish that are not salmonids, salmon, seem to be essentially not sensitive at all. And so when we look at different salmon species, we see that Coho salmon is, the one that's extremely sensitive. But the rest of them are really varied. There's white spotted char and brook trout, which have kind of moderate toxicity, same with rainbow trout and steelhead. But then there's plenty of species like Atlantic salmon, brown trout, where we don't see toxicity at all. And so this is kind of odd for a chemical to not cause similar toxicity in species that are so closely related evolutionarily. But since we don't fully understand the mechanism, we really have no idea why this is happening. So hopefully future studies will help us figure that out. And when they do, I'm going to be really excited about it because this has just been a giant question on so many toxicologists' minds for the last few years. And so that brings us to my research, my PhD research. And the question that I'm really trying to answer is, what are the lethal and sublethal effects of 6-PPD quinone to our fish in California, especially fish that are currently threatened or endangered? And so these are the 5 species that I've been studying. And so I kind of just want to go through and maybe get to know them a little bit. So first we have our coho salmon, which again, very sensitive, started this whole saga. And we decided to include them because they kind of act as a positive control. So we know that if we're seeing similar toxicity in the coho salmon that we're testing in the lab, then our methods are kind of working right, and they're appropriate to actually assess toxicity in these other species that have never been tested before. And also, coho salmon are interesting because they are native to the Sacramento-San Joaquin Delta and the San Francisco Bay. However, they've been extirpated from the bay, meaning they are completely gone for the past couple of decades. Right now in California, the only major population of coho salmon is really far north on the coast. And so we don't really know why exactly the coho salmon are no longer in the bay, but this research is interesting. We can't use this research to confirm that this is why they're no longer there, but it is a big, interesting question. And so next we have steelhead, which are also a very important fish species in California. People fish for them recreationally. And Chinook steelhead have a few different populations that are either threatened or endangered, depending on the population. And then Chinook salmon are also very important to California. They're very commonly fished. And there's quite a few different populations in California, and they're all listed as either threatened or some of them are very highly endangered. And so for these 3 salmon species, they're very ecologically important, economically important to California, and also very culturally important for our California tribes. And so we'll move on to our 2 smelt species. First, we have Delta smelt, which are endemic to the Sacramento-San Joaquin Delta. They don't live anywhere else in the world. And they have been having serious population declines for almost 50 years. And it's to the point where right now they're really straddling the line between endangered and extinct. They have not been listed as extinct, but there have been multiple years in the last decade where we haven't found evidence of a single Delta smelt being present in the Delta. And they used to be the most abundant pelagic little fish in the Delta. And then also we have longfin smelt, which are native to our Delta and have also been having some pretty bad declines. They actually were just listed as endangered a couple months ago, back in August. And so I also want to highlight before I move on that for each of these 5 species that I'm studying, I'm looking at both the larval and the juvenile stages. And that is because the younger organisms tend to be still kind of fully developing their organs, fully developing their detoxification pathways in the liver. And so because they're not fully developed yet, they tend to be the most sensitive to most contaminants. So that's why we're looking at these kind of younger life stages. And so the first thing that I've been doing for each species and life stage that I'm testing is I start by trying to determine that LC50, the lethal concentration. And so here is what kind of that data looks like. This graph was for my larval coho salmon. And so on the x-axis, we have the concentration that the fish were exposed to. So we expose them to a range of concentrations. And then on the y-axis, we have the percent mortality at those concentrations. And so when we plot mortality versus concentration, we're able to get this curve where we can then look at what concentration we would expect to see 50% mortality at. And so that's how we determine the LC50. It's also useful for, you can kind of do the reverse. If you know what concentration you have in the environment, you can try to figure out approximately how much mortality you might expect. And so these LC50s are really common in toxicology, and they're really useful for risk assessments, which are used by environmental managers to figure out how to help maintain these populations. And so here are my results for this kind of chunk of my research. So first, we have coho salmon, which were very sensitive, like we expected. The larvae are actually a little bit more sensitive than the juveniles, which is, again, not uncommon, but this is the first study that's kind of comparing both of those life stages and showing that, yes, it is kind of behaving how we expect in that sense. Steelhead have an LC50 that's pretty significantly higher than coho. The steelhead is at like 2,645 nanograms per liter for larvae, which that concentration is pretty significantly higher than what we see in the environment. However, if you look at the concentration in the environment in the San Francisco Bay and try to look at approximately how much mortality we would expect to see about 7% mortality in steelhead. So still, it's very likely that our steelhead are having lethal responses to the amount of 6 PPD quinone in our streams. And then in Chinook salmon, no toxicity at all. And actually, this says LC50 over 100,000. We actually tested it up to 500,000 and no mortality. They were totally fine. And that was pretty interesting because, and actually unexpected, because there was a study that had just come out, coming out of Canada, the BC area, where they were testing a population of their Chinook salmon, and they were seeing pretty significant mortality at 67,000 nanograms per liter. And that's still way higher than what we see in the environment. But it was interesting because it does kind of suggest that, not only are we seeing differences in susceptibility between these species of salmon, but also there might be differences between genetically distinct populations of the same species. And I guess I will throw out that the population that we studied is the fall run Chinook salmon, which is the most common Chinook salmon that we can find nowadays. So we don't really know for sure what some of the more endangered California runs, what their susceptibility might look like. And then finally, our Delta smelt and longfin smelt, also totally fine, no mortality, which was really great to see because they've been really struggling. And so it's great to see that this isn't, another one of the stressors that we have to worry about for them. And I also want to mention the rest of the data that I'm going to go through is not going to include the smelt, mostly because I haven't finished analyzing the data for them yet. So we're only going to focus on the salmon for the rest of this talk. Okay, so the next kind of test that we do is it's a sublethal effects test. And what that means is that we expose the fish to concentrations that we know are low enough to not cause mortality. And the reason for that is because we can look at effects that you wouldn't be able to see if you have a dead fish, like their tolerance to thermal stress, for example. So this is a thermal tolerance test that we did. And we chose this test because we were seeing those, the gasping in the field of our symptomatic fish are gasping more, their gills are ventilating more. And so we think there might be something happening with their cardiorespiratory system. And the thermal tolerance test is a proxy for cardiorespiratory performance. In this test, after their sublethal exposure, we place the fish into a water bath, like in this picture here, and we separate the fish based on what concentration they were exposed to. And then over the course of a day, we slowly ramp up the temperature until the fish are not feeling great. They're starting to lose their equilibrium. And at that point, we find the fish that's lost its equilibrium, pull it out and put it into a recovery bucket. But we record the temperature and the amount of time that that fish was able to handle the heat stress. And so this is the results that I've seen in our salmon, at least. And on the left, we have an example of kind of one of the graphs that I'm able to generate. This is for steelhead larvae specifically. And so it's a little tricky to read this graph, so hopefully I can do it justice. On the x-axis, we have the amount of time that the fish were able to spend to maintain their equilibrium in that heating water bath. And then on the y-axis, we have the percentage of fish that are still upright. So we see at the beginning of the day, the fish are all fine. The temperature is slowly ramping up. And then at a certain point, they start not feeling great. And then the percentage of fish that are maintaining their equilibrium comes down. So that's what the shape of this curve really means. And so if you look at the black line, that's our control fish. And the highest concentration that we tested is this red line. And so we can see that the fish that were exposed to 6-PPD quinone are not able to handle that heat stress as long, which means it's kind of twofold. They can't spend as much time, and they're probably not able to get up to quite as high of a temperature as the control fish are. And so that's just one example of what the data can look like. But on the right here, we have a table that shows that we do have changes in the thermal tolerance of these fish. In coho salmon, both the larvae and the juveniles, and then only the larvae of steelhead and Chinook salmon. And the numbers that I have in the table are the concentrations where we had that significant change. And so in coho salmon, we see it's a very low concentration, very environmentally relevant concentration. In steelhead and in Chinook salmon, actually, these are supposed to be swapped. Steelhead should be 500 and Chinook should be 100,000. The 500 nanograms per liter is pretty high for environmentally relevant, but we do sometimes see it. So we might expect some effects in steelhead in the wild. And then the 100,000 nanograms per liter, which should be next to Chinook salmon, way higher than what we're seeing in the environment. Okay, and so our next sublethal test that we do is looking at their behavior. So again, we expose the fish to lower concentrations for a few days, and then we put individual fish in bowls, and we use a GoPro basically to record their movements in the bowls over 30 minutes. And this picture here on the right, it was going to be a video and I couldn't get it to work before we started today, but it's I think a little self-explanatory. We have 3 fish here. For my study, I was only doing 1, but we are able to put these videos into animal tracking software. And so this is what that animal tracking looks like. In the video, you would see the fish moving around and that colored tail is like perfectly matching the fish. And so that animal tracking software is really helpful for us because it allows us to analyze different endpoints. So these first 4 here, velocity, how much they're bursting, cruising, freezing, these are all kind of locomotor behaviors. And the last one is an anxiety-like behavior. And so how we can measure anxiety-like behavior is by assessing how much time the fish is spending on the edge of the bowl compared to the center. So normally fish have a certain level of anxiety and they tend to like to hug the side of their container just because it makes them feel a bit safer. But a fish that has less anxiety might be more bold and explore into the center and kind of go all over. And so this software helps us track that as well. So for the locomotor behavior endpoints, we found that locomotor behavior was altered in every species and life stage that we tested. This graph or chart is a lot of data, and I'm not going to get into the nitty-gritty about it, but I want to highlight that every concentration that we tested for coho salmon and every concentration except maybe this highest concentration for steelhead they're all environmentally relevant. So these changes in behavior we do expect to see in the field. And then in Chinook salmon, a lot of our concentrations got really high, but the lowest one that we tested, 10 squared, so 100 nanograms per liter, is environmentally relevant. And so even in Chinook salmon, where we don't see any mortality, we're seeing changes in their behavior. And this is a cause for concern because these behaviors play a fundamental role in a lot of essential biological functions, but the big ones being foraging for food and escaping predation. And so each changes in these behaviors, if they're affecting their ability to find food or escape, it could really influence the survival of an individual and thus the overall health of the population. And behavior was also chosen as an endpoint in this study because in the field, we were seeing the loss of equilibrium, we were seeing the surface swimming and spiraling, and those all suggest that there might be some neurological changes happening. And behavior is a common assay to assess for neurotoxicity. So just because we're seeing the behavior changes doesn't necessarily mean it's neurotoxicity. Mostly we need more info about the mechanism of action before we're really able to make that link. But it is possible. And so that's one of the reasons I'm very excited to see more mechanism of action studies coming out, hopefully soon. And then I also won't get into the nitty-gritty of the anxiety data, but really just the takeaway is that the anxiety-like behavior was altered in all of the species and life stages tested. Most of them became more anxious, so we might expect them to hide more and might not be able to forage for food quite as much. But in steelhead larvae and in Chinook salmon juveniles, the opposite happened. They were more bold and they were more likely to go out and explore. So again, we don't really know how the mechanism works, so we don't really know exactly what's happening there. So in conclusion for my study, there are different susceptibilities across salmonid species. The concentrations found in the environment are potentially lethal to coho salmon and steelhead, very likely for coho salmon. Environmentally relevant concentrations can cause those changes in behavior in every life stage that we've analyzed and species that we analyzed. And then 6-PPD quinone is likely adding additional pressure to these fish populations that are already at risk due to a lot of environmental stressors. And so this is likely just making their situation even more precarious. And so in the future, we're really excited to see those mechanism of action studies, and also to have a risk model developed that incorporates climate change scenarios and potentially other stressors that might interact with 6-PPD quinone and either increase or maybe decrease the toxicity to our aquatic species. And so before I finish, I want to talk a little bit about what we have learned since I started this study. So since I started 2 years ago, there's been a little bit of information about mechanism of action, nothing definitive. But this study, this image here on the left is from a study where they looked at rainbow trout cells. They were looking at gill and liver cells, but I want to focus on the gill cells. And they found that the gill cells had an increased oxygen consumption rate compared to gill cells that had never been exposed to 6-PPD quinone. And that is interesting because, as I've mentioned, there is potentially cardiorespiratory effects happening here. And so we think that that increased oxygen consumption rate in the gill cells is suggesting that there might be disruption to mitochondrial respiration. And so you may remember from high school biology, the mitochondria is the powerhouse of the cell. And so really what that means is the mitochondria is where sugar and oxygen gets converted into a molecule called ATP. And ATP is really important because it is basically the energy that a cell is able to use. So without ATP, a cell will die relatively quickly. And so what we think is happening is that 6-PPD quinone is disrupting that conversion of oxygen and sugar to ATP, because if that happens, we expect to see an increased oxygen consumption rate in those gill cells. They're just trying to compensate for the fact that they're not making ATP. So they're like, come on, give us oxygen, please. Like, let's do this. But the 6-PPD quinone is still really just preventing that from happening. This other study on the right, they were looking at coho salmon that were exposed to runoff, where they knew that runoff causes the mortality syndrome that we were seeing, and comparing that to controls. And what they did in these fish was they injected them with a fluorescent dye in their bloodstream. And what that allowed was for them to create these images, where on the right, we have a control coho salmon, which we can hardly see because it's so dark, which means that the fluorescent dye is not coming through. We're not able to see it in this image. On the left, we have a fish that has been exposed to the roadway runoff and had the dye injected. And we can see that dye infiltrating into the brain region in yellow. And then also the rosette is basically their sniffers. It's really important for salmon and helping them migrate. So it's this dye, really what it's saying is that blood and plasma are leaching into the brain and the rosette. And the blood-brain barrier is a membrane in our brain, around our brain, really, that is supposed to prevent junk in our blood from passing into the brain and causing toxicity. But since we're seeing that fluorescent dye in the brain, it really suggests that blood-brain barrier is compromised. And so that could be potentially part of the reason that we're seeing these neurotoxic-like symptoms. But again, we still have to wait a little bit before we can definitively say that this chemical is acting like a neurotoxin. And so we're going to zoom out even a little bit further and talk a little bit about what we're doing about this. So the California EPA is the first agency to start regulating 6-PPD in tires. The Department of Toxic Substances Control is under the umbrella of the Cal EPA, and they listed 6-PPD as a priority product. And what that means is that the U.S. Tire Manufacturers Association was required to conduct an alternatives analysis. So what that is they were required to look at any chemical that could potentially substitute 6-PPD and still work to protect the rubber from ozone, but also that has less environmental toxicity than 6-PPD quinone. And so they completed the first stage of that alternatives assessment and submitted it to Cal EPA on March 25th of this year. And so when they did that, they looked at 40 different chemicals and they were able to narrow it down to 5 that seem promising. And so right now they are working on stage 2. And so they're looking at those 5 chemicals much more in depth. They're reading all of the literature, trying to figure out anything we can possibly learn about these chemicals. They are doing much more in-depth evaluations of the toxicity and the exposure potential of these chemicals. They're also looking at the toxicity of transformation products. So they're really, really assessing quinone derivatives specifically as well. And then they're also quantifying the economic impacts and what that might look like if we were to make a switch to one of these other chemicals in the future. Okay, and so with that, my presentation is done. I'm happy to take any questions, but I would like to thank Dr. Amelie Segarra, who is the PI on this project. I couldn't have done any of this without her. And everyone in my lab, a lot of them are in this picture, but some of them, we don't have a great picture of all of us. So, but everyone in my lab has been so helpful. It's been a really labor-intensive project. And then on the right here, we also have other PIs from UC Davis that are helping. And finally, I want to thank the California Department of Fish and Wildlife, who provided funding for this project through Prop 1. So yeah, with that, I'm happy to take any questions.
Thank you so much, Jackie.
Yeah, of
Absolutely
course.
amazing. And I love that you were able to make it in a way that many people could understand. So appreciate
I'm
this.
glad.
Yeah.
Yes, that was definitely the goal. And I just think it's such an interesting topic that would interest almost anyone. So I'm so happy to be able to talk about it with people who are maybe not as closely related to the science part of things.
Yeah, for sure. So actually, the first couple comments coming in, I'm sure you can see them, compliments to you. But
Okay.
there's a question here. Have studies been done on the sensitivity of 6-ppdq on lab rats?
Yes, there have been some studies. I keep up with them, but I don't read them in depth because I don't work with rats. But what I can recall is that 6-ppd quinone in rats has been shown to reduce sperm motility. There have been studies looking at 6-ppd and 6-ppd quinone in the lungs because we actually expect that for humans, our exposure to 6-ppd, to both of these chemicals, is going to be primarily through the lungs. And that's because if you remember that slide where I showed you how small the particles are, the small particles are able to be airborne for longer, and they're able to penetrate further into the lung. And so I can't remember exactly what malformations they were seeing in the rat lung tissue, but they were seeing malformations in the lungs. And then one other study, this wasn't in rats, but I found it fascinating. They looked at 6-ppd quinone concentrations in the spinal fluid of people with Parkinson's and compared them to people without Parkinson's. And it was a higher concentration in the people with Parkinson's. So not necessarily a causal relationship there, but also really, really interesting. And I'm excited to see where that research goes.
Yeah, it makes you wonder, doesn't it?
Yeah. Mhm.
Yeah. Next question is, are there similar studies looking at the effects of 6-ppdq in amphibians, particularly tadpoles, since they also breathe through gills?
Yeah, I have seen studies in amphibians, and they did not show any lethality. There probably haven't been many on sublethal effects or maybe like more like abnormalities that aren't going to cause lethality. And I can't remember if I've seen tadpoles specifically, but in amphibians, so far, we haven't seen much to be concerned about.
Interesting. How long lived is 6-ppdq in waterways? Is it broken down by bacteria or in some other way?
Yeah, that's a really good question. So luckily, it breaks down somewhat quickly. The numbers that I've seen are anywhere between like 10 hours and a few days. And so what that means is, in theory, if we are able to find a chemical alternative to 6-ppd, once we make that switch, the 6-ppd quinone concentrations in our ecosystems should go down relatively quickly. And I believe the main way that it's broken down is hydrolysis. So I don't know if there's been a lot of study on how bacteria specifically affect it, but yeah, that is promising. And so that is also something to think about when we're thinking about toxicity is the 6-ppd quinone toxicity is really coming in these big pulses. So not only especially in California, we have a very long dry season where all these tire particles are just accumulating. And then especially in around December, we have that first big rain that flushes it all. And so there's this big pulse of 6-ppd quinone in the waterways. But then after a couple of days, it goes down to basically nothing. So yeah, that is definitely one of the promising aspects of this issue.
Could this be lethal to birds that eat these fish?
Yeah, I don't think I've seen any studies in birds yet. So in theory, it could be, but I'm not really sure. I can't recall a single study in birds, and based on the fact that we really aren't seeing toxicity in many other species, I would say probably not lethal to birds. But again, that's really just a guess on my part.
All right. I am not seeing other questions come in right now. Again, I want to thank you very much for sharing with you, and want to remind all of our people that we're working on December, but we definitely have some great things coming up after the first of the year for Flyway Night. So hope to see you all again. And thank you, Jackie, for sharing your wonderful work, and keep it up.
Of course, thank you. Thank you for inviting me. I'm really glad to be able to talk to you guys today. So, and I'll see you around at some of your future events.
Perfect. Excellent.